This lecture, part 4 of 4, will cover the chemical level of organization in the human body and provide a chemical foundation for the course. This lecture is split into parts A and B. Part A will discuss carbohydrates and lipids. Part B will discuss proteins, nucleic acid, and the compound adenosine triphosphate. Carbohydrates are organic molecules that consist of carbon, hydrogen, and oxygen present in a 1 to 2 to 1 ratio, meaning that for every one carbon atom in the molecule, there will be 2 hydrogen atoms and 1 oxygen atom present. Carbohydrates provide a source of energy for the body and include sugars and starches. There are 3 major types of carbohydrates: monosaccharides, disaccharides, and polysaccharides. Monosaccharides are known as simple sugars and serve as the building blocks for larger carbohydrates. Monosaccharides are water-soluble. They will readily dissolve in water. Glucose, the most important energy source in the body, is an example of a monosaccharide. The chemical structures of glucose are represented below. Glucose can have a straight chain form or a ring form. For those of you with a background in biochemistry or organic chemistry, you may recognize the shorthand form of the simple sugar seen here. Disaccharides are composed of 2 monosaccharides joined together. Disaccharides, like monosaccharides, are also water-soluble. Examples of disaccharides include sucrose, maltose, and lactose. Polysaccharides are composed of multiple monosaccharides and/or disaccharides that are linked together by covalent bonds. Examples of polysaccharides include starch, which is found in grains and potatoes, and glycogen, also known as animal starch. Glycogen, made and stored in the liver and muscles, is composed of many glucose molecules. Glycogen serves as an energy reserve because it can be broken down as needed into glucose, the body's main metabolic fuel. The breakdown of polysaccharides and disaccharides is accomplished through hydrolysis reactions. Recall that in hydrolysis reactions, water is added across a bond, breaking the molecule into smaller components. In this case, the polysaccharide or disaccharide into the smaller components of monosaccharides. The formation or synthesis of polysaccharides and disaccharides is achieved through dehydration synthesis. In a dehydration synthesis, water is removed from 2 smaller components, forming a bond between the 2 smaller components to create the larger molecule. This is seen here. 2 monosaccharides, glucose and fructose, are being combined in the dehydration synthesis to form the disaccharide sucrose. Water will be removed across the 2 individual components of the monosaccharides, will form as a product, and as a result, a bond between the 2 monosaccharides will form, making the disaccharide sucrose. Lipids are organic molecules that contain hydrogen and carbon. The carbon and hydrogen are found in a ratio of 1 to 2, meaning that for every 1 carbon atom in a lipid, there will be 2 hydrogen atoms. Lipids also contain oxygen, but in a much smaller amount compared to carbohydrates. Other elements, such as phosphorus, nitrogen, and sulfur, may also be present in lipids. The category of lipids includes things such as fats, oils, and waxes. It is important to note that most lipids are insoluble in water. Lipids are useful to the body in several ways, including providing long-term energy supplies in the form of fat deposits, and also in forming important structures and features of the cells. Fatty acids are long hydrocarbon chains with carboxyl groups attached at the end. Our carboxyl group is often denoted as a COOH group, sometimes also seen as this. The carboxyl group at the end can readily dissolve in water, whereas the hydrocarbon chain of a fatty acid, the tail of the molecule, is water insoluble. The concept of like dissolves likes can be applied here. Recall that water is a polar molecule. The carboxyl end of a fatty acid is polar and therefore can dissolve in water, whereas the nonpolar hydrocarbon chain of the tail will not be readily dissolved by the polar water molecules. Fatty acids can be either saturated or unsaturated, as seen here. In a saturated fatty acid, all of the bonds between the carbons in the backbone of the molecule are single covalent bonds. Examples of sources of saturated fatty acids include animal fats and butter. Any fatty acid with one or more double covalent bonds between the carbon atoms is considered to be an unsaturated fatty acid. You can see this double covalent bond between two carbons of the carbon backbone in red here. If any double bond is present in the carbon backbone, then the fatty acid is known as unsaturated. If only one double bond is present, it is known as monounsaturated. If more than one double bond is present in the carbon backbone, it is known as polyunsaturated. Examples and sources of unsaturated fatty acids include vegetable oils. Triglycerides. Triglycerides are molecules composed of three fatty acids, shown in blue, bonded to a glycerol molecule, shown in purple. The fatty acids are bonded to glycerol in dehydration synthesis reactions and can be broken off of glycerol through hydrolysis reactions. Triglycerides serve as an energy reserve in the body, providing insulation and cushioning to organs as well, for example, the kidneys. When saturated, they tend to be solid at room temperature. When unsaturated, they tend to be oils at room temperature. Another class of lipids are the steroids. Steroids are characterized by their four connected carbon rings, shown here in blue. Cholesterol, the molecule that's diagrammed below, is the most common steroid. Cholesterol helps maintain cell membranes, and some hormones, including testosterone and estrogen, are cholesterol derivatives. Phospholipids. Phospholipids are found in cell membranes and are composed of glycerol molecule, shown in purple, two fatty acids, shown in blue, a phosphate group, shown in yellow, and a non-lipid group, shown in orange. The non-lipid phosphate group region is water-soluble, and the fatty acid tails are water-insoluble. The next lecture segment will continue the discussion of the important organic molecules of the body and cover proteins, nucleic acids, and high-energy compounds.